That's it. Just one name. Thank you. I heard what you said yesterday.
Well, yes, just short.
Yes, yes.
Knows who you are and has read about you.
Yes. Okay, good. Well, I study the brain. That's my work. That's my main hobby. It's one of my passions. And so what I will talk about is the brain. And if we can show the video, the first part.
Took nearly a half century, but tonight we have a better idea about what made Albert Einstein smarter than the rest of it. It's not about what he learned. Dana's going to tell us it's the shape of his brain. It was extraordinary.
The founder of the theory of relativity died in 1955 at the age of 76. A pathologist in Princeton took Einstein's brain home with him. No major study was ever done on the overall anatomy of Einstein's brain till now. Scientists like Dr. Patrick Gannon at the Mount Sinai School of Medicine are thrilled. Gannon is credited with finding chimpanzees' language areas in their brains, and he calls the Einstein discoveries phenomenal.
This is the parietal region right here, which is what they're talking about in this study. Now, if you look at Einstein's brain, you can see completely different. This is relatively narrow. This is normal human. All the brains I've ever looked at are like this. Einstein's brain was amazingly wide.
In fact, researchers found the part of Einstein's brain related to mathematical reasoning was 15% wider than normal on both sides.
Dr. Gannon went straight to the source earlier tonight and called Dr. Sandra Witelson in Toronto and congratulated her on this breakthrough.
She explained it to me and gave me great detail. He didn't develop it due to his development of his mathematical skills later in life. Chances are he was born with this very unusual anatomy and that's what he inherited at birth.
He was born a genius then.
You could certainly look at it that way.
The scientists at McMaster University compared Einstein's brain with the preserved brains of 35 men and 56 women who had died with normal intelligence. The scientists also found that groove, the Sylvian fissure, did not extend all the way in Einstein's brain.
How do you measure this in our time and in science?
The next obvious step is to do functional imaging, so you will get a genius mathematician, maybe not at the level of Einstein, but a modern-day mathematician, and put them in a functional imaging machine and see which parts of the brain light up. And if it's these regions, and they are expanded compared to a normal non-mathematical genius, then we know that we've hit upon something pretty big here.
Dr. Witelson in Canada found the shape of Einstein's brain is not depicted in any atlas of the human brain. This fascinating study is published in this week's issue of The Lancet, the British medical journal.
So it's nice to have other people doing my work for me. He explained it so well. You've heard a lot about the brain. Although we've had people with so many different areas of expertise, the brain and thinking and cognition have been talked about so much in the last day and a half. So what does the brain do? What my own work has been involved in is not just the anatomy of the brain, but how it supports cognition. What does cognition mean? It's not just one of these sort of dictionary words that refers to just subtle little things that we do. But cognition is really everything. Cognition is when we just discriminate between two simple spheres, different colors. It is perception, it is our memories, it is our feelings, it is our voluntary behavior, and it is our consciousness, which of course is a major topic and probably something for another day. For me, it's been fantastic to be here at this conference, not just to hear the things that have been said, the wonderful ideas, the wonderful images, but as a neuroscientist, I can't stop thinking and seeing things through my own particular perception. And what I'm seeing and hearing are these fantastic brains at work. I mean, what I am actually seeing here and what you are, we're seeing the products of the operation of fantastic, healthy, creative brains. And in fact, what I'm even seeing, I believe, is an experiment. There are some classical experiments in psychology which demonstrate that if you change the visual input a little bit from left to right, skew it, split it, And if you do this in a cat, you could win the Nobel Prize, like David Hubel and Torsten Wiesel did about 10 years ago. You can do it in monkeys. It's now been demonstrated clinically in humans. You actually change the visual system of the brain. So when I see Steve Mann and his students distorting or allowing themselves to see the visual world through split images in a different way, I can only surmise that their visual system is being modified. And I should say that the earlier one does something like that, the more permanent any changes may be. So if your students are out there, I just wanted to let you know. So it's been it's just absolutely amazing to see a potential brain modification in action. So I didn't give Lionel Tiger all my paper, because I knew I was going to need something. So let me now have the first slide, which I think I do. Right, so this is a picture of the kind of picture of the brain that you usually are seeing. It is a computerized image. You can see just roughly the structure. You can see the red areas, the colored areas, which indicate which parts of the brain are actually slurping up the nutrients as the individual is doing some cognitive task. So that's called functional imaging. So with magnetic resonance imaging, we can get magnificent computerized virtual images of the brain. But it's not really the real thing. Here is the real thing in my lab. And this is actually, this demonstrates all these little tools. So I guess it won't go that far, but you can see from the front to the back there is a big cleft, and you can see the outer contour of the brain. That, although it is yellowish, that is what's called the gray matter. You must have heard of gray matter. Those are the brain cells. And then you see the white matter, and you can see the two hemispheres. Now the brain is a particularly complex organ compared to some of our other organs, because it is not homogeneous, but it is functionally heterogeneous. Different parts of the brain do different things. So here you have a schematic, a cartoon of the brain, and the main message here is that different parts of the brain do different things. So for example, if you look at the yellow area, that is crucial. You're using that now. If you're at all paying attention to what I'm saying, you're using this yellow area. When we go out to coffee, a conversation break, you're using this gray area in the front at the bottom, which I can't point to, but it's, believe me, it's that gray area in front of the red bulge. If you look at the back over here where you have the green and particularly the blue area, visual interpretation, that's very important for visual imagery. And I was thinking when I heard Charles Pachter talk about his work and when he said he visually edits his images, I think that's the region that is being used. And that is also the region that one would expect was crucial in Albert Einstein's thinking when he was developing the theory of relativity. He actually, in his own writings, and his biographers have quoted him saying how he doesn't think in words, but he thinks in pictures, he thinks in images. And after he gets the concept, after he would, as he used to say, I will now a little bit think, he would then go and do his thought experiments, and when he had the image and the concept of the space-time way that the universe is put together, he would then talk to mathematicians and put it into words and symbols like E equal mc squared. But the idea came first before the algebraic equations. So, how did I get to have this brain? You could imagine for someone who is interested in studying the brain, to have the opportunity to study the brain of the century and perhaps of the millennium is quite an awesome thing, and it still is. We've had it in our lab now, 1/4 of it in our lab for about 4 years now. So a long time ago, I left McGill University, the security of graduate school of McGill University, went to do some more studying in Manhattan at NYU, and then took a position at McMaster University. And under contract to the National Institutes of Health in Washington, I was contracted to try to figure out what makes that yellow area in the left hemisphere different from the mirror image area on the right side. Because as many of you know, if there is localized damage in that yellow area on the left side, as a mature individual, that individual may become aphasic, lose the ability completely and permanently to speak and to understand language, even though the other side of the brain is perfectly intact. And so my thinking has been, and still is, there must be something at the microscopic level or at the chemical level that is different between the two sides, and if we knew what the difference was, then this would have major clinical possibilities of trying to harness the intact tissue to take over the functions that have been lost. We're still working on that problem after 25 years. Dr. Corin here, who is another neuropsychologist, he knows well that we still don't know, and neuroscience doesn't know the answer. But along the way, we found out some other things. Now, how are we going to do that? Our method was, and is, that we have the cooperation of physicians and then people in the community at Hamilton who have a very poor prognosis for a long life because of metastatic cancer of some organ, but not the brain. And they donate their time so that we do neuropsychological tests to understand their intelligence, memory, musical ability, artistic ability, reading ability, etc. And should they succumb to their illness, they have agreed and their family has agreed, and we get their brains to study when the brain is taken out at a clinical autopsy. So we have a brain bank at McMaster University. So some of the things, and I'll just take a minute to mention this because I don't want to go into details, but this is what the brain looks like from the middle if you cut it in half. You see the white structure, that is the corpus callosum. It has been in the news quite a bit in the last 15 years. There's some suggestion that although every other part of the brain is larger in the male brain than in the female brain, because the overall brain is larger in the male, to perhaps be congruent with the larger body, the callosum in some regions may be larger in the female. But at any rate, what we found is that people who have a left-hand preference have a larger structure. What this structure does is it interconnects the left and the right hemisphere. So it is very crucial for some of the major ways of the brain's functioning. Lionel Tiger mentioned that he talked about gender differences. We all know that there are sex differences in thinking. We know that there are sex differences in behavior. What one doesn't know is what is their cause. You know, it's the question, is it cultural or is it biological? What we found was that this is what the cortex, or that gray matter, looks like. And you can see the symbol on the right-hand side, which is the symbol for female. If you look at it, you don't even need a computer to count it. You can see that there are many more of those little blue dots, which are neurons or brain cells, and the cell packing density, the number of cells per unit volume in the female brain in the language regions is larger. We can't prove at this point that that is the basis of some of the linguistic differences between men and women, but it is tempting to think that this might be one of the differences. So it was this kind of work that was not only in science journals, but also reported in the media, particularly the New York Times, which is, you can't get better than that if you want people to know what you're doing. And as a result of that, in October of 1995, out of the blue, the green fields of Lawrence, Kansas, actually, I received a fax from a very charismatic man in his 90s, Dr. Thomas Harvey. Dr. Thomas Harvey is the pathologist who removed the brain of Albert Einstein in 1955 during a regular, normal clinical autopsy at the request of Einstein's family. And he had been the custodian of that brain, keeping it, as you may have read in various newspapers and magazines over the last 45 years, keeping it in a cider glass jar, in cupboards, in whichever home he had. He did was he was always looking for neuroscientists to do some work on the brain because he himself was not a scientist and didn't have a laboratory. So he sent me a fax and asked if I would like to work on Einstein's brain. And I thought about it for like maybe a nanosecond, and I answered yes. And I have to tell you, my life has not been the same since. There is Albert Einstein, just at the time when he was writing his papers on the theory of relativity. And this is Thomas Harvey in my office with me in January '96. After I said I would like to do this work, he came and he brought the brain. And I can tell you, it is a saga, and I don't have time to tell you everything about how he brought the brain, how he got it across the border, what we did for 2 or 3 weeks, the stories that he told us, and what is still going on a daily basis back home in Hamilton as a result of this. Let me just say, what we did is we were going to do something at the microscopic level, but thought we would do a basic study of the overall anatomy of the brain just as a baseline. And of course, why we were really poised to be able to do this was not that we had any special talents or equipment or anything that anyone else had. In fact, you will see we used a ruler. That was our technical equipment. But what we did have that other people didn't have was a ready-made control group, a baseline group of about 120 brains of people, of men and women who we knew were of normal intelligence. And so that for the first time, here was a brain from a genius that could be compared to a control group. So as you saw in the original video, which they showed, it's the same picture. This is what was published in The Lancet. The top picture shows the brain, Einstein's brain from the top. It is almost circular, which is very unusual. Most of our brains are egg-shaped. The parietal lobes, the overall weight, volume, etc., was normal, within the normal range. But the breadth across the widest part, which is the parietal lobes, was 15% wider on both sides, which meant that that region, which is important for visual imagery, for multidimensional thinking, was larger in this man. The two pictures below, B and C, show the brain from the side, and here the difference is even more compelling. It's not a quantitative difference, but it is a qualitative difference. There is a sulcus, which you will be able to see more clearly in the next slide, because it was done by Time magazine. No, excuse me, well, that's just ours. So here, this is Time magazine that used color, and on the left side, you see a normal brain, and you can see a yellow area. That yellow area is a region that is in front of that black line, which is the Sylvian fissure. And the red area is the parietal region behind the Sylvian fissure, which is just a cleft. On the right-hand side is Einstein's left hemisphere, and as you can see, there is no yellow region. And that is because this cleft or fissure turns up so close to the front of the brain, so anteriorly, that there was no room for that region to develop there. And as a consequence, the parietal lobe was even larger and all situated behind a particular gyrus. So this means the whole layout of his brain was different than any in our collection or that we had seen in any atlas. And this was found in both hemispheres. And so if we could have the next bit of tape, if you don't believe our scientific paper, you will see it in this next clip. Can we have the video?
I'm Dr. Alonzo DeGriff, a medical examiner, retired. I want to tell you how much I admired your article in the July American Scholar.
The Einstein piece.
Brilliant.
Oh, thank you.
Can we talk about it?
Dr. DeGriff, yeah, sure, I'd love to. Why don't you call when I get off at 8 and I'll set up a time?
Oh, no, we have to talk about it now. It's because of what I have in this bag. Oh, it's not a bomb. Well, metaphorically...
Sir, I'm on duty
See, you're
right
the
now.
first person
I'd love to talk to
that
you, but
I
if you
felt
would
I could show this to. It's Einstein's brain.
Would you mind putting that away? Sir, Einstein's brain disappeared after his autopsy.
Yes, I removed it myself during the post-mortem in 1955. I've had it with me ever since, meticulously preserved, of course. So here, take it. Examine it. Find out what made that man tick.
Sir, I'm not a neurologist. I wrote on Einstein and politics.
But you're an Einstein expert. You captured his essence. And you work in the most advanced experimental and research facility we've got. Don't pass this up.
That can't possibly be Einstein's brain.
Well, you've got my card. Check my credentials. You call anyone you think you need to call. You'll see. It's all yours.
No, but I don't. Hey, I've been looking for you. Listen to this. This is gonna knock you out.
I'm already knocked out. I had 2 skull-based craniotomies and a laminectomy today.
So, that brain?
What? Turned out to be Elvis's.
No. Listen. I called the state of New Jersey. They verified that guy, DeGriff, was the ME on the Albert Einstein post-mortem. The cause of death was correct, the date, everything. And I know for a fact that Einstein's brain was missing after the autopsy.
So wait. You're saying this is real?
It just seems to be adding up.
Oh, we could always take a look. Compare it to another brain. I mean, why not?
Mm-hmm.
I mean, if we could find a structural anomaly in Einstein's brain, if we actually have Einstein's brain, we design a study.
You know how Einstein developed his theory of relativity? He pictured himself riding on a beam of light looking back at a frozen clock tower. He had to visualize time as a dimension and the universe as a curved, 4-dimensional space.
My guess, that indicates unusual parietal lobe function. Okay, I'll get a brain from pathology. We'll compare it. We'll do MRIs and...
Dr. Wilkes and Dr. Hanlon.
This is the mystery brain. If you compare its parietal lobes with the brain I picked up from pathology...
They're not divided.
Exactly. I think this short Sylvan fissure is the key. I'm sorry.
Well, I think something went wrong at the very end. But anyways, you get the idea. They did this without calling us, and we're not getting any residuals. But you saw the jargon, the Sylvian fissure, the parietal lobes right in there. So let me close with just mentioning. Two things that I think this kind of work that is going on in neuroscience. I mean, this is just a snippet of it, but you could see you can see the potential and the future that is going on in neuroscience. You know, the brain has been called the last unknown part of the universe. So there are two things. One, you know, to be able to understand how the brain works and to support thinking and feeling is not going to be only of great theoretical importance, but have great clinical implications. And I couldn't help thinking when I heard the compelling words this morning of Norman Jewison, who talked about how wonderful life is, the joys, the excitement, how much there is, and how lucky he is and we are to be alive. But as you well know, there are people who don't have that joy, who can't experience that joy. And they may be in a very similar environment as Mr. Jewison, with the same sexual, professional, family opportunities. But there's something wrong with their brain. And most likely it's the neurotransmitters and some of the anatomical pathways. And those individuals don't find life heaven, but really find it as hell, and sometimes leave. And so the clinical consequences of understanding more about the brain are awesome. The last thought that I want to leave you with is that there are good consequences, but there are potentially dangerous ones. Because if you think about it, if we're going to be able to understand the anatomy and the chemistry of the brain in relation to cognition, we're going to have a new kind of IQ test. Just imagine the scenario when people are going to be applying for a job, or to get into an architectural school, or to get into medical school, and you write for an application, and they're to fill out, and you can imagine that someone's going to say, and send us an MRI scan done on a 4 Tesla machine so we will know exactly what your capabilities are. And this can be, of course, misused and can be difficult. And there are bioethical issues to be dealt with, and they are being dealt with in various groups around the world. So thank you.